Good evening, Professor. This is my personal timetable. So, in the next week, I have planned to complete my 6th task. The first task is PSA biosensor paper writing. As per your suggestion, I have changed in the title and some inner content as well. Also, some modification in the figure 1 with the help of Jaya. And also, I'm just including one heading called selectivity contents in the PSA biosensor paper. And the second task is urine biosensor research. Simultaneously, I'm also working in this task also because to find the better solution for the attachment on the surface, because we already face some technical issues and some biological issues like the better attachment on the surface. So now we found that because of the EDC, so we use NHS as well, so for the better attachment, and as well as the graphene attachment on the surface. So we also clarified with the capacitor measurement of each step, either a antibody and with the antigen binding. So hopefully, and we will deliver the result in this week or in the next week. Task 3 is review paper writing. Is almost 70% is done, but the 30% I'm still working on it, and I'm delivering this paper in this week. And the task 4 is AD biosensor research, that is related with the Halim Medical School, because we got the samples. It's a before TDCS and after TDCS samples. So also working in this task also with the help of also Prashant. So the thing is, the problem is the attachment, because the first initial stage we face the problem. So now we solve it, and we work on this and to solve this issue as soon as possible. So the task 5 is coronavirus biosensor research. So I'm also writing the some research plan and read some literature review for the our related to our research. And the task is AO biosensor research. Sir, as you given me one research article that is related to the meta surface attachment with our research, so I'm also writing a research plan and deliver to you by this Wednesday. So this is task 1, PSA biosensor paper writing. So this is the figure that I have some modification with the help of Jaya, and also need some more modification. So in this week I will send to you figure 1 as a schematic figure, and waiting for your comments and suggestions regarding this. And this one is the task 2, is related with the urine biosensor. So this research is basically depend on two types, either using EDC NHS solution and the APTS. So now we focus on the EDC NHS because it's a better attachment with the APTS, because our chip having a PTTI metal, and we got the better results with the EDC and NHS, so we go with it. And from tomorrow onwards I will immobilize the 3 antibodies with a specific concentration, and then simultaneously attach with the antigen immobilization with a different 5 different concentrations to check via the LCR meter or impedance analyzer. And the responsible person is me, Prashant, and somewhere the Rayon Sang and the Dongmin. So sir, this is the FTIR result that I already did on 26th March from Aju University. So in as per this FTI result, I got this like to EDC having somewhere the void shape we got, but if we combine with the NHS like EDC NHS, then we got the better result than APTS. So we hope for the best and to go with the EDC NHS measurement and the binding affinity. So this is the 3 chemicals that we have: antibody, for the urine biosensor research, and this one is the PSA antigen. So these four samples that we have for the urine biosensor, these three type of antibody and one is antigen. One by one, I just immobilize with one antibody and then one single antigen to check the capacitance value and the comparison with these three antibody, with that this one antigen, and this one is the comparison graph that we have is related with the transmittance and a wave number. So we have the better result with the APTS. As I already told you that if we combine with the NHS with the EDC, then we got the better result than the APTS. The task 3 is review paper writing, and it will be completed and submit to you the draft in this week. Task 4 is the AD biosensor Halim Medic School. So we are also working simultaneously in the lab with the medical samples, and hopefully we will produce the good results in this week. And the task 5 is coronavirus biosensor research. This I'm preparing research plan and send it to you in this week. And the last task is AO meta surface biosensor research. So sir, in this task I have some prepared some research plan and also doing some literature survey, like which one is more suitable for our research and with our substrate, because normally people are using some carbon electrodes and some metallic nanoparticles electrode using different techniques: FRET, electrochemical luminescence, SPIR, colorimetry, SERS. But our case is to just transmittance and transmission method. So just I'm focusing with as per our requirement. So because we have the antibody, antigen, and the AO surface area, and also using graphene oxide as well. So I'm preparing the research plan and send it to you as soon as possible. Thank you, sir.